Hi, my name is Abhishek Bhattacharya. I'm a fourth-year medical student at University of Michigan presenting on single-cell phenotyping using optical imaging and AI. The current method of brain tumor diagnosis requires multiple modalities from hemotoxin and eosin staining of pathology slides to advanced techniques such as whole genome sequencing and methylation assays. However, these can be cost-prohibitive and often be very time-intensive or infeasible in a time-critical environment such as the neurosurgical operating room. Current intraoperative diagnosis requires sending the biopsy sample to the pathology lab, which is then stained and then finally interpreted by a specifically trained neuropathologist. This whole process can take between 30 minutes to 2 hours. Our prior lab work has proposed a streamlined workflow using optical imaging, specifically stimulated Raman microscopy, and AI-based computer vision to make the diagnosis in under a minute. Clinical SRH microscopes are already being used by neurosurgeons at multiple clinical sites. Stimulated Raman spectroscopy works by shining a laser on the sample and then measuring intensities at specific wavelengths that correspond to various biomolecular compositions. The resulting stimulated Raman histology, or SRH image, provides robust contrast of cellular structures without the need for histologic staining. Given the robustness of SRH, can we combine it with AI algorithms to analyze brain tumors at the single-cell spatial resolution and overcome the limitations of single-cell analysis methods? Current single-cell analysis techniques such as RNA-seq have been limited due to loss of spatial information. These methods also scale poorly to hundreds to thousands of samples in a cost-effective manner. Often modern algorithms also aren't being used for intelligent analysis, and most importantly, current single-cell analysis methods are difficult to apply outside of the scientific lab and into the bedside. For this reason, we developed a method for deep cell segmentation of SRH tumor specimens, which includes first imaging the sample using SRH microscope, which provides intensities in two channels. The channels are computationally processed into an RGB image that mimics a virtual H&E stain and divided into smaller patches of 300 by 300 images. These patches are then hand annotated with cell nuclei using a web-based SRH labeler. These are then used to train a faster region-based convolutional neural network and a masked region-based convolutional neural network for automated cell segmentation. The faster RCN model and a masked RCN model were trained on 50 patches that had both a bounding box and a segmentation for each cell, and these were then validated on 1,081 patches that had only a bounding box for each cell. The masked RCN showed a more robust mean average precision, given that it takes both a mask and a bounding box during training time. Empirically, the masked RCN model was consistently able to segment cell morphologies across various tumor types. Here are some examples of various tumors that we were able to segment using our masked RCN model. Thank you for your time and attention for this talk. The work here would not have been possible without the help of my PI, Dr. Todd Holland, and my wonderful colleagues in this lab.